Number 7. What are some examples of processes that might demand high rates of protein synthesis in humans? Growth of hair, growth of skin, replacement of stomach cells, puberty, development, um, any of those kinds of things. Growth of fingernails. Okay, and then using a diagram, summarize the process of translation. That's probably, there's probably a good diagram in your textbook that you can refer to for that. Let's see if I can find it. Yeah, so it's a diagram on 327 and 328. So figure 4 and figure 5 will be the two diagrams for question number 8. Okay, then I asked on page 345, 1, 2, 4, 5, and 8. So let's go there. Page. Okay, question 1. Define the following terms: mutation, frameshift mutation, point mutation, nonsense mutation, missense mutation. Mutation is a change in the genetic sequence. Could be an mRNA, could be an RNA or DNA, but it's a change in the gene sequence or the nucleic acid sequence. Frameshift mutation is when the reading frame, where the codon is read, changes, it shifts. A point mutation is when you change the nucleic acid base at one point in a genetic sequence. A nonsense mutation is when a codon that normally signals or that normally uh, brings in an amino acid changes to a stop codon. And a missense mutation is when one codon uh, changes in such a way that the amino acid that comes in is changed. Okay, the normal form of a gene contains the nucleotide sequence given there. What type of mutation is each of the following, and what effect will this mutation have on the structure of the protein encoded in this gene? 3 extra adenine nitrogen base pairs are inserted in positions 10, 11, and 12. So that's going to be an insertion mutation. And 3 adenines are going to become 3 uracils in the RNA, which means you're going to add a phenylalanine to the code. It will not result in a frame shift because you are adding 3 bases. So you'll reestablish the reading frame. But you would probably get a missense mutation. The third nucleotide, well, no, it wouldn't be a missense. It'd just be a mutation. It's not even a point mutation because it's not happening at one point. It's 3 bases. The third nucleotide changed from an adenine to a thymine. Okay, so ATA becomes ATT, and ATT is UAA. Okay, that's a stop codon. So that's going to be a nonsense mutation. And C, the thymine nucleotide in position 10 is removed. So that's going to be a frame shift mutation. Okay. Question 4. Which of the following amino acids can result from a single base pair substitution, and explain your reasoning? Phenylalanine to leucine. Yes, if you change a UUU or a UUC, which is phenylalanine, to a UUA or a UUG, you'll get that change. So that's a single base pair. So that is correct. B, ILE to theranine. ACU, ACC. You could, yeah, that would work too. So if you look here, right, there's isoleucine, ILE. If the U changes to a C, you go from ILE to theranine, or in the first three cases. Okay. Uh, serine to arginine. Serine is there. Arginine is there. No, can't happen. Not a single base pair, anyways, because you need CG, and we're starting with UC, so you would need to change two bases. And asp to gly. Asp to gly. That could be done with a single base change. The A's get changed to G. Okay, so those are those questions that we looked at yesterday. All right, classroom. Considering it's 9:55, let's say we take a 10-minute break, and then we can um, go on with operons and hopefully wrap up the unit. So go ahead and take. What about the serine and arginine in the same box? Hang on, I missed that one if it's there. Oh, there. Find a proper table. Yeah, I guess so. Yeah, you're right. I missed that pair. So what he's talking about is this box here. That would work as a single base pair substitution. That you could get a Sierra Arch. I was looking at these two boxes. I did miss this box here. So yeah, but then those two boxes would work too. Okay, so I'm gonna go and set up the next PowerPoint. You guys take a 10-minute break. So I'll see you at 10:10. be hopping back and forth between a PowerPoint and a Google slide because some of the animations work better in PowerPoint than they do in Google Slides. But I'm going to start with Google Slides. So where we are, if you look at classwork, did it not come up? Where the hell did it go? Maybe I haven't posted it yet. Hang on. Okay, I guess I haven't created it yet. Okay, here we go. So material, genetics unit, operons, add from Google Drive. Operons, insert, topic, 13th to June 4th, post. Okay, so if you scroll down to May 31st, June 4th, you'll see operons. That's the one we're going to be working on next. I'm going to see if I can find any videos on operons as well. Okay, maybe these guys. Control C, edit. That's the Bozeman one. Regulation is probably better. I wonder if there's a crash course on operons. Looks like not. Okay, so just those two. Okay. Okay, so let's talk about control in bacteria. So this is primarily a bacterial control mechanism. It turns out eukaryotes do have operon systems, but they're significantly different from prokaryotic ones because remember, eukaryotes have introns, whereas prokaryotes do not. Yeah, I know, I just rearranged it now. Okay. Laser pointer. Here we go. Okay, so when we talk about bacteria metabolism, bacteria need to respond quickly to changes in their environment. Okay, so if they have enough of a product, then they need to stop producing it. In other words, if they normally make, let's say, an amino acid, but then they run into a source of that amino acid in the environment that they can just absorb, then they don't need to make it anymore because it'd be a waste of energy to produce more. So how would we stop making more of that product? Stop the production of the enzymes that you need to make the product. Okay, so that's a stop system. Now, what if the other possibility is a go system? If they find a new source of energy that they don't normally use, right? So let's say normally they feed off of glucose, but they manage to find a source of galactose, okay? Then they need to use that galactose quickly, right? Because they need it for metabolism, growth, and reproduction. And so they have to start making the enzymes to digest galactose right away. So normally they don't need those enzymes because normally galactose isn't present in their environment, but now suddenly galactose is present. So they need a go signal that tells them, okay, start making these enzymes because we've got a source of galactose. Okay, when we looked at regulating metabolism, we used feedback inhibition. And we've talked about this before. The product acts as an inhibitor of the first enzyme. Okay, so if we look at the pathway to make the amino acid tryptophan, it starts off as one precursor. Enzyme 1 from gene 1 converts it to product 2. Enzyme 2 from gene 2 converts it to 3. Enzyme 3 from gene 3 converts it to 4. 4 converts it to 5. And 5 converts it finally to tryptophan. Okay, the problem with this is that it's a wasteful production of enzymes. What do I mean? If all of a sudden you land somewhere where there's lots and lots of tryptophan, you don't need to make tryptophan anymore, right? So if you don't need to make tryptophan and you use allosteric inhibition, right? So the tryptophan blocks enzyme 1. The problem then is the cell is still going to make enzyme 2, enzyme 3, enzyme 4, and enzyme 5. And that's a waste of energy because it's enzymes that aren't going to do anything. Because in this system, the gene doesn't know any better to stop making the enzymes because we don't need it. Okay. Now in eukaryotes, this is the way we have to control metabolism. Okay. But in prokaryotes, that's not necessarily true. There is another way. Okay. And that other way, let's move this over here, is by gene regulation. Okay. So instead of blocking enzyme function, so instead of using tryptophan to turn off enzyme number 1, what we're going to do is we're going to block all the genes for making enzymes. So all 5 enzymes will no longer be produced as long as there is a high level of tryptophan. Okay. This saves energy because you're not wasting energy doing protein synthesis for enzymes 1, 2, 3, 4, and 5 when you don't need to because you don't need to make tryptophan. Okay. So how does this work? Okay, gene regulation in bacteria. Every cell is going to vary the amount of specific enzymes by regulating gene transcription. Okay, they're going to turn genes either on or off depending on need. Okay, so the turn genes off example. If a bacteria has enough tryptophan, then it doesn't need to make any more tryptophan. So therefore, it doesn't need the enzymes to build tryptophan so you can turn those genes off. Okay? The turn genes on example, if the bacteria encounters a new energy source, like a new sugar, like lactose, okay, the one you find in milk, then it needs to start making the enzymes so that it can start digesting lactose, which it normally doesn't do. Okay. In that case, you have to have a go system, make the enzymes, and let the system run. Okay. So what bacteria do to run the system is they group genes together. Okay. So this grouping of genes is what we call an operon. Okay. So an operon is a group of genes in a bacteria that have related functions. For example, all the enzyme in a metabolic pathway will be lumped into one operon. Okay. That operon also has a promoter region, just like in eukaryotic cells, right? What we call the Tata box, where the RNA polymerase is going to bind to. Okay. So you have one promoter that controls the transcription of all 5 genes in this metabolic pathway. Right. So there's the promoter site here. And we have something called the operator. It's a binding site for something called a repressor protein. Okay. So the operator site is your on off switch. You turn the operator on, you turn the operon on or off depending on the status of the operator. Okay, it's literally a binary switch. Okay, so how can genes be turned on or off? We use something called the repressor protein. Okay, so it is a specific protein that's going to bind to the DNA at the operator site. So the green yellow portion here, this is the promoter site. Okay, the yellow portion by itself is the operator site. Okay, so somewhere else on the DNA will be the regulator gene. Okay, the regulator gene is going to make the mRNA that's going to make the protein. Okay, now when if this is an off system, then normally the tryptophan, sorry, the repressor protein has a shape that does not allow it to bind to the operator site, right? This shape is wrong for this position, okay? But if there is enough tryptophan in the environment, the tryptophan will bind to the repressor protein, causing an allosteric change, right? So the shape will change so that the repressor will now fit in the operator site. Once it sits in the operator site, RNA polymerase can no longer bind to the promoter. Since it can't bind to the promoter, this gene, which was on, is now turned off. Okay? This is the bit you need to understand how a repressor works. Okay, the next bit I'm going to do on a PowerPoint show rather than as a instead of a Google slide because this works a little better. All right. So how the operon model actually works. Remember that the operon consists of an operator, this red site, the promoter, which is this Tata box here, and then the genes that are going to be controlled. Okay, gene 1, gene 2, gene 3, and gene 4. Normally, let me just turn on the laser pointer again for this thing. Okay, normally, RNA polymerase, if this is a on system, okay, RNA polymerase will attach to the Tata box. Nothing is blocking the operator, so RNA polymerase goes along and reads the genes. It then makes the messenger RNA, okay? And then that messenger RNA will go into the ribosomes, and the ribosomes will make enzymes 1, 2, 3, and 4. Okay? So that's how the system normally works. But, I missed it. But if a repressor is present, so a repressor protein attaches to that operator site, RNA polymerase can no longer attach. Since RNA polymerase can't attach, the mRNA is not made. And since the mRNA is not made, the enzymes aren't made. So the system is now off. Okay? That's the Operon model. That's the basics of how an Operon works. Okay, as I said, there are two types of operons. You have operons that can be turned off or operons that can be turned on. The tryptophan operon is a operon that is turned off. It's normally on, it gets turned off. Okay, therefore we call it a repressible operon. So under normal everyday circumstances in every bacteria, the bacteria do not live in an environment with lots and lots of tryptophan in it. So if it needs tryptophan, it has to make its tryptophan. Since it has to make tryptophan, it has to be making the enzymes to synthesize tryptophan all the time. Therefore, under normal conditions where there is no tryptophan in the environment, RNA polymerase attaches to the promoter site, runs along the entire gene, makes the messenger RNA, and then creates the 5 enzymes required to synthesize tryptophan, and tryptophan will be made. But let's say that this happens to be a very, very lucky uh, bacteria, and the bacteria ends up in a chunk of turkey meat. Okay, turkey meat has lots of tryptophan in it. So when that happens, tryptophan from the environment enters the bacteria. Okay. When that happens, the tryptophan will bind to the receptor because there's lots of tryptophan in the environment, cause the repressor to change shape, and the repressor will now attach to the operator site. So the repressor and its co-repressor, tryptophan, attach to the operator, blocking the Tata box. Since the Tata box is blocked, okay, due to a conformational shape change in the repressor protein, RNA polymerase cannot attach. Therefore, mRNA is not made, and the enzymes aren't made. Tryptophan synthesis stops. Okay? That's a system that is usually on and gets turned off. Repressible operons. Okay. Any questions on that? How long will the repressor protein remain on the operator? Until what happens? Yeah, until there is no more excess tryptophan. When the tryptophan gets used up, right, tryptophan will be released from the co-repressor. When it gets released, the repressor goes back to its original shape. It cannot bind to the operator site anymore, and the system turns back on again. Now let's see if I can find it. Simulations on PHET. What? Don't tell me PHET isn't working. Okay, simulations, biology. Okay, filter. That's a lack operon. What's the gene expression one? Oh, okay, no, that's not the one I want. Search. What do you mean back-end fetch failed? Simulations, browse sims. Which is selection, gene expression, polar, no. Filter. Java. Java via cheap. Subject. Biology. Color vision eating says lack operon. That's one. Natural selection, neuron, pH scale, radioactive dating, MRI, sound, stretching. Okay, so we'll have to do I'll do that one in a minute. Okay, so that's the repressible operon. Okay, so it's normally on. We're turning off. The opposite of that. Normally, what turned what? So the repressible operon is normally on and turned off. What's the other type going to be then? The opposite of it. Yeah, it's going to be normally off and turned on. Okay. So the system that's normally off and gets turned on. Come on. Oh, okay. This is just explain what happens when it's already kind of going over it. So I'll skip that. Is the inducible operon. I like activatable, but the word we actually use is inducible. So it's a digestive pathway. So this is what we call the lack operon. So normally in this system, the repressor is always on the operator because normally we don't want to make these enzymes because normally lactose is not in the environment. So if there's no lactose, there's no point in making the genes that digest lactose. That's kind of pointless. So normally, RNA polymerase can't bind. No mRNA is made, no enzymes are made because there's no lactose in the environment. But if suddenly E. coli bacteria ends up in your milk, okay, or in ice cream or yogurt, whatever, and there's plenty of lactose now suddenly available in the environment, the lactose is going to attach to the repressor, cause the repressor to change shape. It's then going to fall off. Since the repressor is removed, RNA polymerase can go. It'll make the mRNA and thereby make the enzymes to digest lactose until lactose levels go down such that the repressor reattaches. Okay. So normally the um, repressor protein has a shape that allows it to lock into the promoter site. But if lactose is the protein, and the protein can no longer bind, and the lac operon turns on. Okay. All right. We're going to show you this. So this is the lac operon. Once it runs. Come on. Way slower than normal. Okay. So let's install everything. The lac I gene goes there. The lac Z gene goes there. Um, this is the lac I promoter. This is the lac promoter. This is the lactose operator. Where's the injector? There it is. So that is the repressor protein, right? And when the repressor protein is on the um, lac operator, there's your lac, um, the enzyme that's going to digest lactose. There's no lactose around, so nothing's going to happen, right? So the polymerase can't go. This thing just continuously makes more and more promoter or repressor. Okay. Now, watch what happens when I start adding lactose. The repressor changes shape. It can no longer bind here. So it's found a lot of lactose. Okay. So the lac RNA polymerase can now run along the gene and start making the lac digesting enzymes. Now watch what happens to the levels of lactose as the enzymes start getting to work. Run this a little faster. Okay, lactose levels have now dropped sufficiently. But the repressor protein changes back, locks onto the promoter site. Synthesis of the digesting enzyme is no longer made, and the system shuts down. Okay, so that's the way that system works. Uh, where's my slideshow? Okay. Now, okay, so this system of operons, okay, was discovered by Jacob and Monod. I don't think that's how you actually pronounce his name, but I don't know how to say it properly. And they named this the lac operon. So that's what was found first. The tryptophan operon, the TRP operon came second. They were the first to describe the operon system, and they coined the actual phrase operon. Okay, so it consists of two parts. Okay, you've got the regulator gene, which makes the repressor protein, and then you've got the operon itself, which is going to be the promoter, the operator, plus all the genes that make up the system. Okay, so operon summary. Repressible operons usually function in anabolic pathways, so digestion, or sorry, buildup, synthesis, synthesizing end products. And so when the end product is present in excess, the cell allocates resources to other uses. Oops. All right. Ah, thought there was another symbol coming up. So that's what we call the off system. The inducible operon normally functions in catabolic pathways, digestive ones. Okay? Produce enzymes only when the nutrient is available. So the cell avoids making proteins that have nothing to do, and the cell allocates resources to other uses. So that's a go system. All right, it's 47. That's basically it. That finishes this unit. Um, and we're done for the day. So I will see you guys tomorrow. You are dismissed. Take care. Bye.